I am Payal Priyadarshini, PGT Biotechnology, Kendriya Vidyalaya number 3, Delhi Cant. Today I am going to make you learn about RNA, its structure and function. In my last session I explained about DNA, the different composition of the DNA, the structure of the DNA and the function of DNA. First let us recall all those concepts, then we will move ahead about RNA. As I already explained in my last session, DNA is a nucleic acid which is composed of nucleotides. Nucleotides is a combination of nucleoside and the phosphate molecule, and nucleoside is a combination of sugar and nitrogenous bases. So, there are three main components of nucleic acid. One is sugar, second one is nitrogenous bases, and third one is phosphate. DNA is considered as genetic material in all living organisms except some viruses. Why DNA is considered as genetic material? Genetic material means the chemical compound which carry the genetic information, the traits, the characters from one generation to another. That genetic information is nothing, those are the protein molecules. The DNA is being forwarded from one generation to another generation, and DNA by the process of transcription and translation synthesizes various types of protein which are being expressed in our body system. So, let us know about RNA, its structure and function. So, RNA is ribonucleic acid. What is nucleic acid? Let us understand in this way. RNA is nucleic acid. Nucleic acid means what? Nucleic acid means the polynucleotide chain. Polynucleotide chain means when many nucleotides combines together, they form polynucleotide chain which form nucleic acid. Now, what is this nucleotide? Nucleotide, when one nucleoside combined with one phosphate molecule, they form one nucleotide. Now, what is this nucleoside? Nucleoside is combination of sugar and nitrogenous bases. Now, if you can see, these are the three main components of the nucleic acid: sugar, nitrogenous bases and the phosphate molecule. These three components make the nucleic acid. Now, let us move towards what are the sugars which are present in the nucleic acid, especially in RNA. So, if we talk about sugar which is present in RNA is ribose sugar. And that is why RNA is considered as ribonucleic acid, that is RNA. Because of the presence of the ribose sugar in the nucleic acid, the RNA considered as ribonucleic acid, and the ribose sugar is one of the type of carbohydrate. Now, what is this carbohydrate? Carbohydrate is polyhydroxyaldehyde and polyhydroxyketone. What is the meaning of polyhydroxyaldehyde and polyhydroxyketone? Polyhydroxyaldehyde and polyhydroxyketone means any organic compound which contain aldehyde and more than one hydroxyl group is called polyhydroxy aldehyde or polyhydroxy ketone. If you can see in this diagram, this organic compound is having polyhydroxy, that is alcohol group, and aldehyde group. This one is aldehyde and these are alcohol group. These aldehyde and alcohol make organic compound carbohydrate, and the carbohydrate which is present in RNA is ribose sugar, which have 5 carbon, 1, 2, 3, 4 and 5. But this ribose sugar, you can see in the diagram, even in the slide, this ribose sugar is found in linear structure. But if we talk about the chemical nature of the sugar, all the sugars, all the carbohydrates are found in cyclic structure. Now question arises, why the carbohydrate found in cyclic structure? The reason, the answer is formation of hemiacetal or hemiketal. Hemiacetal means whenever any aldehyde or alcohol, this one is aldehyde and this one is alcohol. So whenever any aldehyde or alcohol interact with each other, they form hemiacetal. Similarly, whenever any ketone reacts with any alcohol, they form hemiketal. So let us have a look how the reaction goes. The hydrogen of alcohol transfer over the oxygen of carbonyl carbon and it make OH. And as we know, the carbon is having 4 valency, so the 4th valency will be satisfied by the remaining part of the alcohol. Remaining part of the alcohol means OR dash. Now this OR dash will satisfy the 4th valency of the carbon, and the formation of this compound is called hemiacetal. I am repeating once again because it is very much important to understand the structure of the sugar molecule. Whenever any aldehyde reacts with alcohol, it forms hemiacetal. In this particular reaction, the hydrogen of alcohol will transfer over the oxygen, it will form hydroxyl group, that is OH, and the 4th valency of the carbon will be satisfied by the remaining part of the alcohol, and they will satisfy the remaining part, that is 4th valency of the alcohol. Now this particular compound is called hemiacetal. The same fashion of the reaction occurs in the carbohydrate, because carbohydrate is also having aldehyde as well as alcohol group. Let us see how the reaction goes in the carbohydrate. So this one is a ribose sugar, which I am going to make it here. Ribose sugar is having 5 carbon, that is why it is known as pentose sugar. Let us get number of this carbon, 1 carbon, 2 carbon, 3, 4 and 5. So this is the ribose sugar. The 4th number of carbon hydroxyl group, the hydrogen will transfer over the oxygen of aldehyde and it will make aldehyde into alcohol. And as that of the formation of hemiacetal and hemiketal, the 4th valency of this carbon will be satisfied by the remaining part of the alcohol, and that is why the sugars, the carbohydrates are found in cyclic form. This particular structure was given by Fischer. This structure can also be written as this way. If we talk about the number of carbon, here the first carbon, second, third, fourth and fifth. So, this particular carbohydrate is a ribose sugar which is found in RNA. Now, if we talk about nitrogenous bases which is found in RNA, there are two type of nitrogenous bases. One is purine and another is pyrimidine. Purine again have been classified into two categories. One is adenine and another is guanine, and pyrimidine is of three types: cytosine, thymine and uracil. In case of RNA, there is no thymine in the RNA, uracil is there. So, out of 5 nitrogenous bases, only 4 are present, and those are adenine, cytosine, guanine and uracil. You can see the diagram of all these nitrogenous bases in the slide. This particular molecule is adenine, this one is guanine, this one is cytosine and this one is uracil, and this particular nitrogenous base is thymine, which is not found in the RNA, it is found in the DNA. If you can see the structure of these nitrogenous bases, you can find that when the methyl group is being added with this uracil, it will converted into thymine, and when the amine group is being removed from this cytosine, it will get converted into uracil. The detail of this we will discuss in the coming slide. Let us move ahead. Phosphate molecule. As I told you, the nitrogenous bases, the sugar and the phosphate molecule, these three main components combines to form one nucleotide, and many nucleotides combine to form nucleic acid, and this nucleic acid is of two types. One is DNA and another is RNA. In DNA the sugar is deoxyribose sugar, and in RNA the sugar is ribose sugar. The basic difference in between these two nucleic acid is the presence of ribose sugar and deoxyribose sugar. Now, let us see how the nucleosides are being synthesized inside the cell. As we know, nucleoside is a combination of sugar and nitrogenous bases. Sugar in case of RNA, it is a ribose sugar, and in my earlier session, earlier slide, I have explained you about the sugar. This is the sugar molecule which is a ribose sugar. Now, question is how this nitrogenous base combine with the sugar to form the nucleoside. You have already seen the structure of the nitrogenous bases. This is a particular one of the typical structure of the pyrimidine. Here you can see this is the NH group, the hydrogen of this NH and the OH of the sugar molecule combines and they will form water molecule. The water molecule will release from here and the remaining part of the nitrogenous base will bind over the first number of carbon. So, in our nucleoside, nitrogenous base always bind at the first number of carbon. This is how the sugar and nitrogenous base binds together to form nucleoside. Now, if we talk about nucleoside, let us move towards the nucleotide. As I have already explained about nucleotide, nucleotide is a combination of nucleoside and the phosphate molecule. Nucleoside is combination of sugar this is a sugar molecule. Here the nitrogenous base will bind, I explained you in my last class, and at this particular 5 carbon the phosphate molecule will bind. The hydrogen of the phosphate will react with the alcohol of the sugar, it will form water and the remaining phosphate will bind over the 5th number of carbon. So, in a particular nucleotide at the first number of carbon nitrogenous base will always attach and at the 5th number of carbon the phosphate will get attached and this will form one molecule of nucleotide. So, let us summarize all this concept. There is sugar molecule in nucleotide, there is nitrogenous base at the first number of carbon and there is phosphate molecule at the 5th number of carbon. So, this is one nucleotide. In this particular compound you can see at the first number of carbon nitrogenous base is there, at the 5th number of carbon phosphate molecule is there and this one is the sugar molecule which is called as backbone of the nucleic acid. So, this is a complete nucleotide. Now, how the nucleotides combines together, how they form polynucleotide chain and how they constitute nucleic acid. Let us have a look. Formation of polynucleotides. This is a sugar molecule. Here we have nitrogenous base at the first number of carbon. Here is the alcohol, second number of carbon, alcohol at 3rd number of carbon, so this one is one nucleotide. Let us draw the another nucleotide so that we can understand how two nucleotides combines together. So, this is the another nucleotide, the OH of the first nucleotide and the hydrogen of the second nucleotide, they will combine together and they will form water, the water will get released out and the remaining part will combine together and they will form phosphodiester bond. Let us have a look how the phosphodiester bond is being created in between the nucleotides. this one is the first number of nucleotide, the second nucleotide, so this is how the two nucleotides combines, and this is called phosphodiester bond. Again I am explaining this bond. The two nucleotides combines together, there is reaction between the alcohol of the third number of carbon of the first nucleotide and the fifth number of phosphate molecule. There is release of water molecule and there is oxygen bridge between the two nucleotides, and they will form phosphodiester bond like this. This is how the polynucleotides are being synthesized inside the cells, how the nucleic acids are being synthesized inside the cell, how the phosphodiester bond is being created. Now let us have a look about the structure of the RNA. So what I explained earlier, this you can see in the slide. This one is a one nucleotide, another nucleotide and another nucleotide. Like this all the nucleotides combines together with the help of phosphodiester bond, and they will form a linear structure which is called the primary structure of the RNA. But few of the RNA also form hairpin structure because of the complementary sequences in their strands, and because of the complementary sequences they form hairpin like structure, and this is called secondary structure, which is found in ribosomal RNA and transfer RNA. This is the basic difference between RNA and DNA. This I already explained in my last session of DNA. DNA is double stranded, you can see in the slide. DNA is having guanine, thymine, cytosine and adenine as nitrogenous bases, while RNA is having uracil, guanine, adenine and cytosine as nitrogenous bases. So a basic difference between these two nucleic acid are DNA is double stranded, RNA is single stranded. DNA is having GTCA as nitrogenous base, while RNA is having UGAC as nitrogenous base, and in spite of uracil DNA is having thymine, and in spite of thymine RNA is having uracil. So this is the basic difference between DNA and RNA. If we talk about sugar, in DNA there is deoxyribose sugar and in RNA there is ribose sugar, which I explained in my last slides. The difference in the deoxyribose sugar and ribose sugar is at the second number of carbon in DNA there is deficiency of one oxygen, that is why this particular sugar is called deoxyribose sugar, and in case of RNA there is hydroxyl group, there is OH group, that is why this particular sugar is called ribose sugar and they are pentose sugar. Now these are the four types of triphosphates nucleotides which makes the RNA, but there are some uncommon nucleotides which are found in the RNA. The one is pseudouridine, which is found in transfer RNA. Pseudouridine, when the third number nitrogen and fifth number carbon rotate at their position by 180 degree, the position of the molecules get changed and the bonding of hydrogen get increased. In the uracil there is 2 hydrogen bonding, while in pseudouridine there are 3 places where the hydrogen bond can be take place. The next uncommon nucleotide is ribothymidine. In spite of uracil, the thymine nitrogenous base get attached with the sugar and they will form ribothymidine, which is found in RNA. Another uncommon nucleotide is dihydrouridine. You can see in the slide in uracil nitrogenous base, this unsaturated bond will be broken down by the addition of hydrogen and this unsaturated bond will be converted into saturated bond, and the addition of 2 hydrogen at these places makes the uracil dihydrouridine, which is found in transfer RNA as well as in ribosomal RNA. The next uncommon nucleotide which is found in RNA is inosine. Here you can see with the sugar, hypoxanthine molecules get attached at the first number of carbon instead of other nitrogenous bases, and they form inosine, which is found in transfer RNA in the anticodon loop, which we will see in our next slide. So, these are the types of RNA. Basically, there are 3 types of RNA: messenger RNA, it is written as mRNA, transfer RNA, that is tRNA, and ribosomal RNA, that is rRNA. Beside these 3 main RNAs, other RNAs are also there in our cells: a small nuclear RNA, micro RNA, a small interfering RNA, a small nucleolar RNA, and catalytic RNA, which is also called ribozymes. Let us have a look what are these RNA and what are their functions. Starting with messenger RNA, as we know, messenger RNA used as template to make proteins. Why messenger RNA is used as template to make protein? What is there in the messenger RNA which helps the cells to make the protein? This is a typical diagram of central dogma, you can see in the slide. This can be explained here also. DNA, by the process of transcription, makes all the type of RNA. The formation of RNA from DNA is called transcription. Here all the types of RNA is being synthesized by the process of transcription. It may be a messenger RNA, it may be transfer RNA, and it may be ribosomal RNA. Now, particularly messenger RNA will codes for amino acid, and they will form another macromolecule of the cell, which is called protein, and this particular flow chart, this particular reaction is called central dogma. So, central dogma is the transfer of information from DNA to RNA and then RNA to protein. The transfer of information, the transfer of traits, the transfer of characters, the formation of RNA from DNA is called transcription, and again the RNA, particularly messenger RNA, will codes for the protein by the process of translation, and they will synthesize the protein, and this whole flow chart is called central dogma. This is how the proteins are synthesized in our cells. The director is DNA, through the RNA they synthesize the protein, and this is called the genetic information which is carried out by the DNA. So, this is the function of messenger RNA. Messenger RNA codes for amino acid, the amino acids combines together with the help of peptide bond, they form the protein, and protein perform various functions in our cells in our body. Moving to next, transfer RNA. You can see transfer RNA, this is a diagram, a picture of clover leaf. If we can see, this is transfer RNA and clover leaf, there is similarity in their outline. The transfer RNA appears like clover leaf, that is why this is considered as clover leaf model of transfer RNA. The structure of the transfer RNA appears like clover leaf. Here you can see this particular loop is called anticodon. This anticodon will bind with the messenger RNA during the protein synthesis and help in formation of protein. This particular loop is called dihydrouridine loop or arm, which I already explained in my earlier slides. Dihydrouridine is formed because of the addition of 2 hydrogen at the unsaturated bond, and this one is pseudouridine arm, which I also explained what are pseudouridine. This is an uncommon nucleotide present in the RNA, and this is formed because of the 180 rotation of N3 and C5 carbon atom, and this arm is called amino acid linking arm. At this particular arm, the amino acid is being attached over here during the protein synthesis. So, this is how the transfer RNA looks inside the cell. Function of transfer RNA, as I explained earlier also, transfer RNA helps the messenger RNA to make the protein. Whenever the signals are given by the messenger RNA, whenever the messenger RNA gives the signal, gives the codes for the protein, the particular transfer RNA will bring a particular type of amino acid during the protein synthesis. The detail of this we will discuss in the coming sessions. Next to that, ribosomal RNA. Ribosomal RNA is having secondary structure. It is not a linear structure. They have various loops, various hairpin structure, and these are the base pairing because of the complementary sequences. The function of ribosomal RNA is to synthesize ribosomes, and these ribosomes help in protein synthesis. There are some another RNAs also. The first one is a small nuclear RNA which help in RNA splicing. RNA splicing means what? In case of eukaryotes, do you know what are eukaryotes? Eukaryotes are those organisms, those cells which have a defined nucleus, which have well organized cells, which have well organized cell organelles. So, eukaryotes are having a DNA with functional and non-functional sequences, functional and non-functional sequences. These functional and non-functional sequences are considered, functional sequences are considered as exon, and non-functional sequences are called introns. When this particular DNA undergo transcription, they will synthesize messenger RNA, and this messenger RNA is called pre-messenger RNA. This pre-messenger RNA contains functional and non-functional sequences, that is exon and introns, and by the process of splicing and processing, these non-functional sequences are being removed and the functional sequences are being added up. The process is called processing and splicing. By the process of this, all these non-functional sequences of the messenger RNA will get removed. The detail of this we will discuss in the coming session, and all the functional part will get attached with each other to form mature messenger RNA. And now this mature messenger RNA will undergo translation to synthesize the protein. In the process of splicing and processing, this particular small nuclear RNA will help so that the mature messenger RNA can be synthesized. Next to that, we have microRNA. MicroRNA, these are the small regulatory RNAs that are approximately 20 to 26 nucleotide in length. The existence of miRNA and their function in gene regulation. Gene regulation means whenever we need a particular type of protein, a particular type of enzyme, the gene undergo their expression, the gene undergo transcription and translation, they will synthesize protein. But whenever we do not need any protein, we do not require any enzymes, the gene has to stop their function. To regulate the function of gene, miRNA help in that. Next to microRNA, we have a small interfering RNA which also help in regulating the gene expression, and nowadays many of the experiments, researches are going on a small interfering RNA to control the expression of any defective RNA so that the defective proteins cannot be synthesized and the person can be cured from the disease. A small nucleolar RNA which are found in nucleolus of the cell, and it help in the processing of ribosomal RNA. The last but not the least, catalytic RNA. Catalytic RNA means the RNA which catalyze certain biochemical reaction, and this catalytic RNA is called ribozyme. This ribozyme will catalyze certain reaction. It do not require any protein, any enzymes for any activities. That is why they are considered as catalytic RNA. Now, in my last session I asked you few of the question. Let us answer those question. The first question in my last session was, why thymine instead of uracil in DNA? As I explained you, in DNA there are 4 nitrogenous bases: adenine, thymine, cytosine and guanine, and in RNA there is uracil, cytosine, guanine and adenine. So, question arises, a very important question, why DNA is having DNA is having thymine instead of uracil. So, if you can see, I also explained in my earlier classes the structure of uracil. This is a particular structure of uracil. This one is first number of atom, second, third, fourth, fifth, and this one is sixth one. This is a particular uracil component. Because of the addition of methyl group, this uracil get converted into first number of carbon, second, third, fourth, fifth and sixth. You can see this is a thymine nitrogenous base. A major difference between uracil and thymine is the presence of methyl group. By the addition of methyl group in the uracil converted into thymine, and this is the reason why DNA is having thymine, because if we talk about base pairing, thymine only binds with the adenine, while uracil bind with adenine as well as the other nitrogenous bases, even itself also. So, due to course of evolution, the uracil changed into thymine by the addition of CH3, so that the stability can be provided to the genetic material, and so thymine is there in the DNA, not the uracil. The another reason is the cytosine can deaminate to produce uracil. Let us see how the reaction happens. This is a cytosine, this particular cytosine when undergo deamination, that is removal of ammonia group, it will become uracil. So, it will be very difficult for the repair system in the cells to recognize the original uracil or the uracil originated from the cytosine. So, these are the two reasons why DNA is having thymine instead of uracil. The another question which I asked is, which evolve first, DNA or RNA? If we talk about the stability, the DNA is more stable than that of the RNA. Why DNA is more stable? Because of the sugar molecule, which I am going to make you explain in my coming slide. And because of the stability which the evolution given us, that is why we can say the RNA evolved first and then the DNA. The DNA originated from the RNA. Let us have a look in the another question. Which one is more stable, DNA or RNA? As I told you, DNA is having deoxyribose sugar and RNA is having ribose sugar. The sugar in the DNA makes the DNA more stable than that of the RNA. Why? The sugar which is there in the RNA is likely to have digested, hydrolyzed by the molecules. Let us have a look what happens when a particular ribose sugar undergo nucleophilic attack. Here is a nitrogenous base, here is a OH group, and this is how the phosphodiester bond is being created. This one is 5th number of carbon, 4th, 3, 2, 1. Let us draw another nitrogenous nucleotide. This is the nitrogenous base, OH group, and this is the phosphodiester bond. Whenever there is addition of water molecule, this particular oxygen will react with here and they will form water, and this phosphate molecule will form a ring structure, and this is the reason why this ribose sugar is more unstable, because this alcohol will participate in the reaction, it will hydrolyze the phosphate group and they will form a ring structure. Now let us answer this question. My first question is, what are the differences between DNA and RNA, which I have already explained in my last session of DNA, and in this session also there are many differences in DNA and RNA, you can answer yourself. The another question, what will be the sequence of this DNA molecule? If you have been given this DNA strand, what will be the RNA sequence of this DNA molecule? This you need to answer yourself, I will explain in my next session. And third question is, give the function of following RNA. What will be the function of messenger RNA? What is the function of transfer RNA? And what is the function of ribosomal RNA? That is all of today's lesson. We will meet again in my next session in the continuation of with this.